Hello, and welcome to Lecture 4 of our course on stochastic modeling of biological processes. In this lecture, we will start with our discussion of Chapter 2, which is focusing on differences between deterministic and stochastic modeling of chemical reaction systems. Chapter 2 will be discussed in 4 videos entitled Lectures 4, 5, 6, and 7. This lecture, called Lecture 4, will cover Section 2.1, while the following lectures 5, 6, and 7 will cover the remaining sections of Chapter 2. In these 4 lectures, we will discuss a number of examples of chemical reaction systems. To develop and analyze their stochastic models, we will use methods which we learned in Chapter 1, including the Gillespie algorithm and the chemical master equation. In Chapter 1, we have already introduced several examples of chemical reaction systems to explain these methodologies. However, for all examples presented in Chapter 1, the deterministic OD-based models provided a reasonable description of the average behavior of the system. In Chapter 2, we will show that the difference between deterministic OD-based models and the average behavior of stochastic models can be significant. We will present chemical systems which cannot be modeled by ODEs, and we will discuss questions which cannot be answered by ODEs. Our starting model in section 2.1 is written for one chemical species called A, which is subject to the following 4 chemical reactions. The third and fourth reactions are the degradation and production reactions, which we have already studied in chapter 1. We added these 2 reactions. 3A goes to 2A and 2A goes back to 3A. In summary, we have 2 reactions which add molecules of A to the system and 2 reactions which remove them. The classic deterministic model is written for the concentration of chemical species A, which we denote by lowercase letter a. It is an ordinary differential equation which has 4 terms on the right-hand side. The 2 positive terms correspond to 2 reactions which add molecules of A to the system, and the 2 negative terms correspond to 2 reactions which remove molecules of A from the system. Since we want to compare the deterministic model with the stochastic description of our chemical system, we need to rewrite the ordinary differential equation to 2 in terms of the number of molecules of A, which can be obtained by multiplying the concentration, the lowercase a, by the system volume nu. Therefore, we multiply the ODE to 2 by the system volume nu to obtain the following ODE for our deterministic approximation of the number of molecules. Equation 2.3 has a cubic polynomial on the right-hand side, which includes 4 unknown parameters. The behavior of this equation will depend on its parameters. In the lecture notes, I specified their values, and the resulting cubic polynomial on the right-hand side had 3 roots. We left it to you to verify in exercise 2.1 that this ODE has 3 steady states for our parameter values. 2 of them are stable, equal to 100 and 400, and there is an unstable steady state 220 between them. In particular, the long-time behavior of solutions to this ODE will depend on the corresponding initial condition. Choosing 4 different initial conditions, I plotted the corresponding solutions in figure 2.1 on page 34. If the initial condition is below the unstable steady state 220, then the solution of the ODE 2.3 converges to the stable steady state 100 for large times. On the other hand, if the initial condition is above the unstable state 220, then the solution of the ODE 2.3 converges to the stable steady state 400 as time t goes to infinity. We want to compare the deterministic description with the corresponding stochastic model, which can be simulated using the Gillespie algorithm, given as the SSA A5D5 on page 25. To apply the Gillespie algorithm, we need to specify the propensity functions of each reaction. We can find them in tables 1.1 and 1.2 on page 16. We have already seen the propensity functions of production, degradation, and bimolecular reactions. The propensity function of the trimolecular reaction 3A goes to 2A is proportional to the number of available triplets, which is proportional to A times A minus 1 times A minus 2. Having specified the propensity functions, we can apply the Gillespie algorithm. Starting with no molecules in the system at time 0, one realization of the Gillespie algorithm is plotted here as the blue line. The red line is the solution of the ODE, which we have already seen in the previous figure. We use the same parameter values in both deterministic and stochastic models, and we calculate the time evolution of the system for 2 minutes. We observe that the stochastic model looks like a noisy solution of the ODE, which was the behavior that we reported for our examples of chemical systems in Chapter 1. Next, we will continue our simulation for a longer time. Let us look how both solutions will continue if we increase the time interval of 2 minutes to 100 minutes. The result is given in figure 2.2 on page 36. The first 2 minutes of these simulations were plotted on the previous slide. You can see that for the initial 20 minutes, the number of molecules of A continues to fluctuate around 100. But then the number of molecules dramatically increases and starts to fluctuate around 400. The stochastic model is able to switch between the two favorable states of the system. It switches back and forth between 100 and 400. This phenomena cannot be described by the deterministic model. If we analyze the ODE, then we can find the steady states, but the solution of the ODE converges to one of them and stays there, as you can see here with the red line. In particular, if we want to find out what is the average time to switch between the favorable states of the system, we have to use a stochastic model. In addition to one stochastic realization plotted here, we can also use the Gillespie algorithm to calculate the corresponding stationary distribution, which we plot here as the yellow histogram. You can practice such calculations in exercise 2.3 on page 56. Since model 2.1 is a very simple chemical system, we can also easily obtain the stationary distribution from the chemical master equation. It is given as formula 2.8, which is plotted here as this blue line. The comparison is excellent. There is no visible error between the yellow histogram and the blue line. In exercise 2.3 on page 56, you are asked to obtain similar results for other parameter values. Let me note that if the results of your computer code do not match the theory, as in this plot, then it could indicate that there could be a mistake in your computer code. A possible error, which students sometimes make, could be in the calculation of the stationary distribution. To estimate it using the Gillespie algorithm, you have to record the values of the numbers of molecules at equidistant time intervals. This is not the same as recording the value of A after every reaction event, because reaction events happen more often when the system is in the state with 400 molecules than in the state with 100 molecules. Therefore, we should not be saving the number of molecules after each iteration of the Gillespie algorithm. Having explained the simple illustrative chemical system 2.1, I will conclude my lecture 4 with a slightly more complicated biological model. This example is a system of two mutually repressing genes, which is discussed in more detail in the paper by Kepler and Elston, reference at the end of section 2.1. I have drawn it here as a schematic with some nice colors because I want to explain some biology which is behind this example. A gene is a region of DNA that encodes function. In this example, I have two genes. I will call them gene 1 and gene 2. These genes can be expressed and make the corresponding proteins, which I have denoted as protein 1 and protein 2. They are pictured here schematically as these circled numbers. Those of you who studied some biology before will know the central dogma of molecular biology, which states that DNA is first transcribed into RNA, which is then translated into proteins. Put differently, this gene makes another molecule called RNA, which then makes the final protein. However, in this illustrative example, both transcription and translation are simplified into this one arrow. So for some people, this will still be a simplified cartoon example, but it has all essential components which are needed to illustrate our main points. In our model, gene 1 produces protein 1. While we only have one copy of a gene in the system, there can be many of these proteins. Some of them will react according to this bimolecular reaction. This is actually a reaction which we have already studied. Using our previous chemical reaction notation, we would denote the numbers of molecules of protein 1 as one chemical species, say A, and the number of dimer molecules as another chemical species, say B, and we would write this reaction as A plus A goes to B. However, this picture better illustrates what is going on here. Two proteins just stick together to create the dimer. The dimer molecule can then bind to the operator region of gene 2. An operator is a genetic sequence which allows this dimer to attach to DNA. In my model, this dimer acts as a repressor protein. When it binds to the operator of gene 2, it switches off the production of the corresponding protein. This is very important. Not all proteins are needed all the time, so gene expression needs to be tightly regulated. In fact, if something goes wrong with the gene regulation, we could end up with some nasty diseases, like cancer. In my example, the dimer molecule of protein 1 can bind to the operator region of gene 2 and it switches off the production of protein 2. This reaction can go both ways. The dimer can also unbind and the production of protein 2 is resumed. In particular, if there is a lot of protein 1 in the system, then there will be also a lot of dimers available to switch off gene 2. So protein 2 will be produced less often. If gene 2 is switched on, then it produces the corresponding protein 2, which can also dimerize according to this bimolecular reaction. And the dimer of protein 2 can bind to the operator region of gene 1, where it again acts as a repressor. If the dimer of protein 2 is bound to the operator of gene 1, then it switches off the production of protein 1. In particular, we obtain a system of two mutually repressing genes, which is a common motif in larger models of gene regulatory networks, which can include tens or hundreds of different types of genes and proteins. The last two reactions included in our model are the degradation reactions for each protein. Having explained how this system works, we could rewrite it as a chemical reaction network using the notation from our lecture notes. It will be a system of 12 chemical reactions, which I've highlighted in my picture by labeling their rate constants as K1, K2, K3, K4, K5, all the way to K12. Let us now count a number of chemical species. We will have to keep track of the numbers of molecules of protein 1, dimer 1, protein 2, and dimer 2. So these are already 4 variables. The other 4 variables are gene 1 with a dimer bound to it, gene 2, and gene 2 with a dimer bound to it. In total, we have 8 chemical species. However, the chemical species corresponding to genes will only take molecular counts of 0 or 1. So we could also describe the state of the system with only 6 independent variables, which would keep track of the protein numbers and the states of genes that can be either on or off. In any case, once we rewrite my schematic as the chemical reaction system, we can use the Gillespie algorithm to simulate it. I am presenting a result of such a simulation here. In this simulation, I am calculating the numbers of molecules of protein 1 and protein 2. You can see that the number of molecules of protein 1 fluctuates around the value of 500, but then it switches to fluctuates around the value of 1000. So we observe that this model qualitatively behaves like our initial example. It has two favorable states, and the stochastic model is capable of describing switching between these two favorable states. Looking at the time evolution of the second protein, we also confirm that our genes are mutually repressing each other. If you have a larger number of molecules of the first protein, like for example around time 100, then the second gene is more likely to be switched off, so we have less molecules of the second protein in the system, and vice versa. So we can conclude that the stochastic description of this model can switch between two favorable states, at least for the parameter values considered in our plots. I have formulated this model using 12 parameters, 12 rate constants K1, K2, K3, K4 until K12. However, it is often the case of mathematical modeling in biology that we do not have accurate information about the values of some of our parameters. So a natural next question is, what happens if we change our parameter values? To illustrate what type of behavior this model can have, I will first reduce the number of parameters from 12 to 6 by making the model symmetric. That is, I will assume that both genes are produced with the same rate, both proteins are degraded with the same rate, and so on. Therefore, this symmetric model has 6 parameters. To get some insights into the behavior of this model, I will fix the values of 5 of these parameters, K2, K3, K4, K5, and K6, and I will vary the value of the production rate K1. I will assume that all parameters have already been non-dimensionalized, so that I don't need to worry about physical units. Our illustrative results are plotted here for different values of the production rate K1. I plot the time evolution of the number of molecules of protein 1 and protein 2 over the time intervals of the same length. My previous illustrative results were for K1 equal to 1.14. So these two plots have already been discussed before. If I increase the production rate K1, then the two favorable states of the system are more separated, and it is more difficult to switch between them. The average time to switch between the states increases. If I further increase K1, then the observed time evolution did not switch at all during the time window considered. I would have to increase the time window to see some switching. On the other hand, if I decrease the production rate K1, then the two favorable states will be closer to each other, and it will be easier to switch between them. So the average time to switch between the favorable states will decrease. Here I included the result for K1 even smaller, for K1 equal to 0.8, when there is only one steady state of the ODE model, and the stochastic system just fluctuates around it. As you can see in these plots, both protein numbers fluctuate around the value 600. Obviously, the results presented here are the results of one realization of the Gillespie algorithm for each parameter value. We can use them to get some insights into the model behavior, but such results will depend on the sequence of random numbers generated by our computer codes. So the next natural question is, how does the stationary distribution depend on the model parameters? Because the stationary distribution is a reproducible, predictable quantity, which can give us a lot of information about the system. We could also analyze the corresponding deterministic model to confirm that it has one stable steady state for smaller values of k1 and two stable steady states for larger values of k1. Another question is, how does the mean switching time depend on the model parameters? Such questions can be asked for many models, including our starting simple chemical system 2.1. You can further investigate the system 2.1 in exercise 2.3 on page 56. We will also discuss some additional approaches to answer these questions in chapter 3 and apply them to the chemical system 2.1 in section 3.8. The gene regulatory network model is not discussed in the lecture notes. In Oxford, our course is also taken by beginning graduate students, who are assessed by working on small projects, which extend the material from the lecture notes. Some of them have written their essays on stochastic models of gene regulatory networks, including the above example. This brings me to the end of lecture 4, where we have discussed section 2.1 covering stochastic modeling of systems with multiple favorable states. Please read pages 33 to 37 of the lecture notes before watching the video of lecture 5. In our next 3 lectures, we will continue in our discussion of chapter 2, which uses examples of chemical reaction systems to highlight the main differences between deterministic and stochastic modeling. In particular, in lecture 5, we will discuss the behavior of stochastic models close to the bifurcation points of deterministic ODEs. Thank you very much for listening to Lecture 4 of Oxford course on stochastic modeling of biological processes. Bye bye.